Okay, let's dive in. Think about this for a second. You've got life's instruction manuals, DNA and RNA. They use almost the same alphabet, A, G, C, but then one uses T, the other uses U. Seems tiny, right? But what if that one little letter swap is actually fundamental, like a key to how life works? Welcome to the deep dive. We take the sources you provide, and today we're looking at some really interesting material, drawing insights inspired by works like Genome Compass. We dig into these sources to pull out the insights hidden inside. Today we're tackling those core molecules, DNA and RNA, and zooming in on that specific difference. Why thymine, T for DNA, and uracil, U for RNA. Our mission here is to understand why. Why this isn't just some random chemical quirk, but actually a brilliant evolutionary move. A design choice, shaping how life stores its info, how it uses it, and even how it protects it. It fits nicely with how some authors view genomics, looking at fundamental mechanisms to understand bigger pictures. Yeah, think of it like this, sort of like the source suggests. DNA is the master composer, writing this grand symphony meant to last forever.
Really, the permanent score.
Exactly. And RNA. That's like the ensemble of improvising musicians in the cell.
The players reading sections of the score, bringing it to life right now, adapting.
Performing in the moment, they've got different gigs, and their chemistry, as we'll see, is perfectly tuned for those roles.
Okay, so let's set the stage. The building blocks. DNA, RNA, they're both nucleic acids, essential for, carrying genetic information.
In everything alive.
Right. They're polymers, long chains made of repeating units. Nucleotides.
And each nucleotide has 3 parts: sugar, phosphate, and the base.
Exactly. And the bases are the letters, A, G, C. They carry the actual information sequence.
And they share most of them. Adenine A, guanine G, cytosine C, those are common to both.
A pairs with T or U, G always pairs with C standard rules.
But then we hit that key difference, the 4th base.
Wow.
DNA uses thymine T.
And RNA uses uracil, U. And you look at them chemically, they're really, really similar.
Almost identical.
Very close. Both pyrimidines, single ring structures. The only difference is this little tag on thymine.
A methyl group. C.
Precisely. Thymine has it, uracil doesn't. Just a small cluster of carbon and hydrogen atoms.
Okay, a single methyl group. It really does seem like such a minor detail. How does that one little addition cause this huge split in function?
Well, that's the beauty of it, isn't it? That small tag actually has significant effects. It subtly changes the shape and stability of how the bases pair up, those hydrogen bonds holding DNA together.
Okay, affects stability.
But maybe even more importantly, it makes thymine more resistant to certain kinds of chemical damage that just sort of happen in the cell spontaneously.
Damage resistance and stability. Okay, now that sounds crucial for DNA's job.
Exactly right. DNA is the archive, the master blueprint. It has to be copied super accurately and passed down generation after generation.
For potentially millions, billions of years.
Yeah, the integrity of that information is absolutely paramount. Errors, mutations in DNA, they can stick around. They can become permanent.
With potentially huge consequences.
Which brings us to a major chemical headache the cell has to deal with. A weakness in one of the bases they both share.
Which one?
Cytosine. C. It turns out cytosine is, prone to a chemical reaction called deamination. It happens spontaneously.
Deamination sounds like losing an amino group.
That's exactly what it is. An amino group gets clipped off the cytosine base.
And when that happens to a cytosine, what does it become?
It becomes uracil.
Uracil, the RNA base inside DNA.
Yep. It just happens. A C in your DNA can naturally, spontaneously flip into a U.
Whoa. Okay, hold on. So a letter that's supposed to be a C can just morph into a U inside the DNA blueprint.
It can. Now, think about the problem this creates. Imagine just for a second that DNA normally used uracil, just like RNA. So the DNA alphabet was A, G, C, U.
Okay.
If a cytosine deaminated and turned into a uracil,
Yeah.
how would the cell know?
It wouldn't. If U is a normal letter, how would it tell if this U was supposed to be there or if it was actually a damaged C?
Exactly. It would be completely indistinguishable. A damaged C would just look like a regular U.
That sounds like a recipe for disaster for keeping the code accurate.
It's a nightmare for error correction.
Mhm.
If the cell didn't spot that uracil as damaged, what happens next time the DNA copies itself?
The replication machinery would read the U?
Think it's normal and put an adenine and A opposite it.
Right, because A pairs with U.
Then, in the next round of copying, that new A would pair with a T.
So, wait. You started with the CG pair?
And after 2 rounds of replication, because of that one undetected C to U switch, you end up with a TA pair permanently.
A C permanently mutated into a T just like that. And the
Yeah.
cell wouldn't even know.
Exactly, a silent mutation. And these would just build up over time, degrading the genetic information. Not good.
Okay, so how did life fix this? What's the evolutionary hack here?
It's so clever. Life made a choice. It decided that DNA would only use thymine, T, as its standard fourth base.
It ditched you entirely from the normal DNA alphabet.
Right. And by doing that, it made any uracil found in DNA an instant red flag.
Yeah.
It always means something's wrong.
So U in DNA is basically a universal signal for damage here, specifically a damaged cytosine.
Precisely. Because U doesn't belong in normal DNA, the cell has repair crews, enzymes like uracil DNA glycosylase UDG.
Okay.
These UDGs are constantly scanning the DNA. If they bump into a uracil, they know it's wrong.
So they cut it out?
They snip out the wrong base, yeah. And then other enzymes come in to patch it up, putting the correct cytosine back. It's an active repair system.
That is incredibly elegant. So using T isn't just about T being slightly more stable itself.
Although it helps.
Right, but the bigger deal is that not using U as a standard base creates this built-in proofreading system for C damage.
Exactly. DNA's choice of T is a sophisticated strategy to maintain accuracy over immense timescales. It's error detection built right into the code structure.
It's a fantastic example of optimizing for that long-term stability, that archival function, which, again, ties into that broader perspective of understanding how these deep molecular choices shape biology.
Okay, that totally makes sense for DNA, the reliable archive.
Yeah.
But then, why does RNA bother with uracil? If T is so great for stability and repair, why didn't RNA just adopt T as well? Yeah, good question. It comes back to RNA's different job. The improvising musician, remember?
Right, the temporary worker.
RNA molecules, mRNA, tRNA, RNA, they do specific jobs right now.
Yeah.
Carrying messages, building proteins. They aren't meant to last forever.
An mRNA molecule might only be around for, what, minutes, hours?
Exactly. It's made, used, and then often quickly broken down. It's disposable.
So the extreme accuracy needed for DNA replication isn't quite as critical for a short-lived RNA message.
It's less critical, yes. An error in one temporary RNA molecule usually isn't catastrophic like a permanent DNA mutation. The cell makes lots of RNA copies.
Okay, so accuracy is less of a pressure. What's the advantage of U then? Why actively choose U for RNA?
Efficiency. It really boils down to metabolism and energy cost.
Making T costs more.
It does. Synthesizing thymine, because of that extra methyl group, requires more biochemical steps and uses more cellular energy than making uracil.
Like a more complicated assembly line in the cell's factory.
That's a good way to put it. Uracil is simpler, quicker, and cheaper for the cell to produce.
And since RNA needs to be made quickly, often in large amounts for immediate tasks,
Using the metabolically cheaper U makes a lot of sense. It saves energy and resources.
it's a molecular cost-benefit analysis. DNA pays the higher price for T because it needs the stability and repair check for its permanent archive role.
While RNA takes the cheaper, faster option, U, because it's all about speed, efficiency, and temporary function. Disposable utility.
So TVSU isn't just chemistry, it's this fundamental evolutionary division of labor baked right into the molecules.
Absolutely. DNA, precise, stable, long-term storage, using T for that vital repair advantage. RNA, rapid, efficient, temporary action, using the cheaper U.
It's this beautiful molecular partnership. Two molecules, almost identical, but that one small difference equips them perfectly for completely different but complementary jobs.
Preserving the score versus playing the music.
And it all comes back to that one tiny methyl group on thymine.
It does. But, the story of that methyl group gets even more interesting when you look beyond just TVSU.
Oh.
Well, remember cytosine C. We talked about how it can deaminate to U.
Right.
In many organisms, humans included, cytosine itself can get a methyl group added to it, creating something called 5-methylcytosine.
Methylation. That's epigenetics, right.
Yeah.
Changing gene function without changing the DNA sequence itself.
Exactly. DNA methylation is huge. It helps control which genes are turned on or off, influences development, and it can be affected by environment, experience.
Which fits that theme of gene-environment interactions being key.
Right. Now, here's the twist. This 5-methylcytosine can also deaminate, just like regular cytosine.
Okay.
But because it already has that methyl group, when it deaminates, it doesn't turn into uracil. It turns directly into thymine.
Wait, methylated C turns straight into T, the normal DNA base.
That's right, spontaneously.
But wouldn't that be harder for the cell to spot as an error? We just establish U as the red flag in DNA.
Precisely the problem. The C to U change makes an illegal base, U, which the UDG enzyme spots easily. But the 5-methyl C to T change, it creates a normal DNA base, T.
So the repair enzyme scanning for weird bases might just miss it. They see a T where a C should be, but T looks perfectly fine in DNA.
Much harder to catch efficiently. And as a result, these C to T mutations, especially happening at these methylated cytosine sites, are actually one of the most common types of single-letter mutations in our genome.
Wow. And the source material links these specific C to T mutations to bigger issues.
Very much so. They're heavily implicated in things we're all interested in, like how some cancers develop and maybe even aspects of the aging process itself.
So this methyl group, it's
Yeah.
central to making T work for DNA stability and C to U repair.
Yes.
But it's also involved via methylated C in creating these hard-to-fix C to T mutations linked to disease and aging. That
Yeah.
sounds complex, like a double-edged sword.
That's a really good way to think about it. It's not just a structural piece. That methyl group is acting like a high-level switch, influencing stability, error checking, gene control via epigenetics, and even contributing to mutations that drive disease and evolution.
It really scales up from a tiny chemical detail to cell function, health, even how species change over time. It underscores that idea of fundamental mechanisms having broad impact.
Absolutely. It's a molecular detail with massive downstream consequences.
Which brings us back beautifully to that analogy. DNA, the composer, needs that robust, error-correcting T for its timeless score.
And RNA, the improvising musician, needs the quick, cheap U for its performance in the moment.
One built for permanence, the other for transience. It really is an amazing piece of natural design.
Stunning optimization, really.
So wrapping up this deep dive, the key takeaway is that this T versus U difference, that single methyl group, it's not minor at all. It's this fundamental evolutionary choice, a trade-off, really, balancing the absolute need for DNA's accuracy and stability
Enabled by T's role in flagging C damage,
against RNA's need for speed, efficiency, and, well, disposability.
which the simpler, chipper U provides.
It shows how life builds these incredibly critical functions, things that affect everything from inheritance to aging, on these really elegant, sometimes subtle, molecular solutions. Tiny details, huge consequences.
Evolution optimizing across different pressures.
Yeah, and it leaves you wondering, doesn't it? Especially thinking about how genes interact with our world. How many other little chemical tags or structural quirks are out there in our biology? Things we're maybe just starting to figure out that are playing equally vital roles, maybe shaping gene expression, our health, our complexity in ways we haven't even fully grasped yet.
That's the exciting frontier, isn't it?
Yeah,
Always more layers to uncover.
a really powerful thought to end on. And that's our deep dive for today.